We're very excited to introduce Dr. Shahram Akbarian. Dr. Akbarian is professor of psychiatry and professor of neuroscience at the Icahn School of Medicine at Mount Sinai. Dr. Akbarian's work explores epigenetic regulation of gene expression in order to identify epigenetic drug targets and novel treatment avenues for psychosis, depression, and other psychiatric disease. We're all very excited to have you, Dr. Akbarian. Thanks, and the floor is yours.
Hi, thanks the whole entire ARIMA team for giving me the opportunity to talk. And first of all, let me double check. Can you hear me and can you see my slide?
We hear you loud and clear, and I think if you put that slide presentation into presentation mode, we'll be good to go.
Okay, so now I press slideshow. Do you now see?
There you go.
Perfect, good. All right, good. And I guess the video camera doesn't work. I mean, I don't mind to show my face, but somehow I clicked on it and it doesn't seem to be activated, but that's fine. As long as you can hear me, everything is good.
We're good to go. Thank you.
Good. Okay, so I have about 20 slides, and I think the first 7 slides are science, and the last 12 or 13 slides are technology based. So let me very quickly, as academic investigators, we're driven by the science. So I explain to you first what is the focus of our laboratory and how we use Hi-C in order to study the genomic architecture in the human brain. And we, as neuropsychiatrists, are limited to working with human postmortem brain, so it has its own limitations, and we often have grants that require sometimes hundreds of brains to be processed in fairly consistent manner. So some of the data you see today are probably not the most technically, not the latest hotshot technique, but it's more based on, the protocol working reliably so we can process many brains. So, so we started with some of our first ARIMA projects. We started with a medical problem that can be summarized as follows. So you have a lot of psychiatric diseases where the patients have, such as schizophrenia, for example, where the patient has a lot of medical comorbidity, like diabetes, hyperlipidemia, excess body mass, which is not a trivial problem. It contributes to the terrible 10 to 20 year reduction in life expectancy gaps for these patients. And this medical comorbidity is very hard to study on a genomic level because the genetic risk architecture of psychosis, including schizophrenia and metabolic syndrome, which summarizes the term of diabetes, hyperlipidemia, and obesity-related syndrome complex, that there is very little overlap in terms of GWAS-based risk polymorphisms, et cetera, et cetera. So it's kind of, a typical situation where you have population level, very relevant medical comorbidity, but you have very little to study on the level of genetics. So we thought, to make a long story short, maybe we should test the hypothesis that lack of this shared GWAS-based genetic risk architecture does not necessarily preclude that there is no genomic interaction or shared chromosomal environment, including chromosomal conformations, in the nucleus of brain cells that are critically involved in the regulation of cognition and the regulation of metabolism. And there are some cell populations in the brain who have kind of double or triple duty, who are multifunctional, and one of them is, as many of you probably know, the midbrain population of dopaminergic neurons, which I try to express on this slide. So to the right, it shows, can you see my mouse? Do you guys see the mouse?
Yes, we can.
Okay, so you see this dopaminergic neurons project to the not only to the brain's reward circuitry, but also to the hypothalamic centers, which are critical for metabolism and nutrition. And on the left of this Zoom screen is the evidence summarized that probably everybody knows about it as well. that links the dopaminergic neurons, both the neurons and its postsynaptic receptor systems, to the pharmacology of psychosis. So anyway, so we said, let's start out and let's examine this if we find something in terms of shared chromosomal environment of these, of these genetic risk architecture. So we had to develop a FAC sorting protocol for midbrain dopaminergic neurons, which works fairly well. To make a long story short, when you deal with postmortem brain, the nucleus as a physical entity is fairly well preserved, even 6, 20, sometimes 30 hours after death. It is a much more stable entity, so to speak, in the postmortem tissue as compared to synapses and neuronal processes, etc. So this nuclei can be even from frozen brain, because they hold together by their chromatin. They can be FAC sorted after immunotagging. We do this with dopaminergic neuron-specific transcription factors. Then we run a certain quality control like nuclear RNA-seq, and this shows some of these tracks. The red is from this FAC sorted midbrain dopaminergic neuron nuclei. Here is TH on the bottom right of this Zoom screen, right? Tyrosine, where my mouse is, tyrosine hydroxylase, clearly showing very nice enrichment for these Nur1+, Nurn+, these are our dopamine neurons, and much less signal for some of these, from these other cell populations. So we kind of developed this assay, and the only problem then for us was that we often got from a postmortem brain only 5 or 10,000 of these nuclei sorted. So it doesn't work very well with run-of-the-mill conventional high-C protocols, which, as you all know, require millions of nuclei as input. So this was a bit of a challenge, and we spent, I think, more than a year on figuring this out. And I will show you later in my slides the protocols that worked. And this slide just, the upper right is classical high-C interaction matrix, TAT, topological associated domain landscape, is really only there to convince you from the data that the protocol works, that we are able to get nice high-C data out of these very limited numbers, 5 to 10 or maximally 5 to 50,000 nuclei at best, input material from this postmortem brain. So we're very delighted. And then the rest is fairly straightforward. We looked for topological associated domains, chromatin domains, that even if they don't have overlapping genetic risk sequences, risk polymorphisms for excess body mass index or BMI and schizophrenia, we looked for topological domains that have somewhere in the domain risk polymorphisms for one conditions, and another portion of that same domain risk polymorphisms for, in this case, schizophrenia. So, and by doing that, we then put the whole high-C data set into this Chrome 3D genome modeling algorithms that has been developed a couple of years ago, I think by a group in Scandinavia, and it works fairly well. And what you see here on the upper, and this was work done by Sergio Espeso-Gil, what you see here on the upper left, this multicolor, is each color is a chromosome. So this is basically the Chrome 3D solution after 1 million or so Monte Carlo based iterations of this solution of the of Chrome 3D, how these topological asset domains are arranged. Each of these beads, each of these little beads is one domain, and this resolution is still not ideal. It's not the same fine resolution as with if you have an input material of 1 or 2 million nuclei, but it's good enough. It was good enough for this purpose. And then what the bottom left shows in red are each of these red beads is a chromatin domain that harbors both risk polymorphisms for schizophrenia and risk polymorphisms for excess body mass in that same domain. And then what Sergio did, he did some Euclidean distancing measurements and found out, much to his delight, that there are at least 10 locations inside these nuclei of these dopaminergic neurons where chromatin domains which share these risk variants for these both conditions seem to form kind of what he calls Euclidean hotspots, basically a non-random clustering of multiple domains, sometimes from different chromosomes, that seem to basically coalesce in a non-random manner. And when he looked for the type of genes that are expressed in these domains. This is in the upper right of these Zoom slides. There is a number of very interesting signaling pathways, brain-derived neurotrophic factor, BDNF, kinase signaling pathways. When you looked for, this is on the bottom right of this slide, when you looked for transcription factor motifs, there was enrichment for dopaminergic neurogenesis, white fat cell differentiation, and so on. So this was all quite exciting, and for us, one of the most striking examples of the promise of this technique were certain Euclidean hotspots, where I'm jumping in the interest of time because I want to talk about the technique. One of these neighborhoods of risk-associated chromatin domains included sequences on chromosome 16, chromosome 17, and 22. And what the cool thing here is that it captured a well-known locus on chromosome 16p, which harbors copy number variants, where the patients are in some ways psychotic or otherwise cognitively impaired, and they're all either abnormally, they have either basically abnormally thin or abnormally sick. So they have always disturbances in body mass, and it can go in both directions. So it's a very interesting and mysterious locus on chromosome 16p, and much to our delight, our High-C assay identified that this locus is heavily interconnected, or has a lot of, I should say, spatial proximity in this nuclei of these dopamine neurons to loci on chromosome 17 and 22, where master regulators, master transcription factors for cholesterol biosynthesis and triglyceride biosynthesis are located. And these transcription factors are well known in the metabolic field under SREBF1 and SREBF2. So this is the kind of example, I think, or promise of using High-C data to map spatial topography of chromatin domains inside the nucleus and develop new hypotheses along the lines I just mentioned. So I don't want to go too much into more of this science, and I want to emphasize that by now we have, I think, a fairly good handle on how to do High-C in these postmortem tissues within this context I just explained. And we basically have two favorite protocols for two very different applications. We developed one protocol, both of these protocols are based with ARIMA High-C kits by now because they're very convenient for us to use. And I'm saying this, I'm not paid by ARIMA or anything, so is that right? I don't have a conflict of interest. It's just happened that they're for us quite straightforward to use in the lab. And so the top protocol is based on this, this basically what I just have shown you as in this first part of the talk. This is this more challenging protocol where we FACS sort nuclei from very small brain regions, such as the ventral midbrain. And so this only involves a small number of nuclei, 5 to 50,000 nuclei at best, limited input material. The other protocol is more standard, where we take larger chunks of brain tissue, such as, for example, from the prefrontal cortex, and then use this material, which basically includes 1 or 3 up to 3 million of nuclei, to do the standard High-C protocol. So all these protocols involve, at the beginning, have a very similar path, cross-linking, lysis. In case of these dopaminergic neurons, we have to do a FACS sorting in between, but the big differences come in the next few steps. So we have modified a little bit of what has been described as to do from what has been described as to do in the ARIMA High-C kits, and Marina and others from our team are very happy to provide you with some more technical details. I'm skipping a little bit over this slide because I think the key differences are here, and we come back to that later. So in what we basically do is we increase, when we generate the libraries from these kits, we change a little bit the cycle number. So we increase the PCR, number of PCR cycles when we prepare the libraries to 14 cycles for the low input protocol, and keep it to 10 cycles for the larger numbers of nuclei. And what it boils down to is, much to our delight, when we send these for sequencing, first by MiSeq, and then if the MiSeq looks good, we sequence deeper with the Illumina NovaSeq system in these days. So the cis-trans ratio is even for the low input protocol with a small number of FACS-ordered nuclei, still pretty good, actually. I have to say. It's up to 50 or sometimes 75% of all chimeric products are in cis, so from the same chromosome. This is a very rough quality index, so the cis trans ratio is pretty good as expected, 2 or 3 to 1, both for the midbrain and for the homogeneous samples, and same thing for the overall number of chimeric reads. The big difference is in terms of things that you normally don't want to see, like PCR duplicates, dangling ends, etc., etc. So when you look here on the right of this, this is a big difference, right? On the right of this slide for the prefrontal tissue homogenate, you see that most of these sequences that come back from the sequencer are of really high quality, and there's a very little PCR duplicates, etc., which basically this is what you would expect from a standard Hi-C protocol. While if you use our low input Hi-C protocol on the left, you see that the number of duplicates and other artifacts is definitely higher than for the conventional protocol, but it's still, you know, for us good enough to generate high quality Hi-C maps in general from these, even with this material. And so this shows just some readout in terms of cell type specific separation between dopaminergic and non-dopaminergic neurons. I think I can go over this because in the interest of time, it's now already 20 minutes, and you know, so this is probably the last slide, just to summarize the principal difference in working with homogeneous and millions of nuclei versus limited samples with just a very much smaller number of nuclei. But both protocols work, and we can basically recommend and happily to share both protocols with the wider field. And with that, I say thank you for listening.
All right, thank you very much. One question here for you is, I think regarding, you know, I think generally you hear less about interchromosomal types of interactions, but you showed that example on your slide of those interchromosomal links between the domains that have both metabolic and schizophrenia risk variants. What do you think would be some of the underlying mechanism of that co-association in the nucleus?
Yeah, that's a good question. I think in general, I think there are two answers. One is, if we take a regular Hi-C data set and we ask for transchromosomal contacts, the only way that we can define them reliably is if we have very large window sizes. So if we have, for example, 1 megabase wide bins, you can, even at very high confidence, call transchromosomal contacts, but that's not of interest for the majority of questions, right? So what happens is, what we did in our case, we first did, as I said, this Chrome 3D genome modeling that uses Monte Carlo iteration and some basic parameters from chromosome polymer physics, I believe, to come up with a solution of having the closest distance without breaking the chain, you know, of these TAT interactions. And once you have the Chrome 3D model for the internuclear topography of all of the TATs in the genome, and once you have that, you define these Euclidean hotspots and say, this is now a group of TATs that are closer together than you would expect from a random model. And then within that group of TATs, you look for these transchromosomal contacts. So it is not anymore against the genome-wide background. And that would be the best answer I can offer. I should also say that this is, in my opinion, only a first step of further, you know, developing the model by doing, for example, DNA fish and other experiments to validate the spatial proximity of these TATs and any locus to locus transchromosomal contacts within this cluster of TADs from these different chromosomes.
Great, thank you very much. One other question here from from Dan from the virtual audience is, could you please comment more on the TN5 high C technique that you mentioned on one of your slides, please?
Yeah, so that's a good catch. And I, again, in the interest of time, I didn't want to talk about it. So TN5 high C was initially a protocol that we developed in-house to accomplish this challenge of generating high C maps from small numbers, a few thousands of human brain extracted nuclei. It basically was a protocol that is entirely based on restriction, digestion, ligation, and transposase-based fractionation of the chromatin without biotinylation. And it was a protocol that gave us, I mean, enough material that we published this genome medicine paper and some of these TAD maps, Chrome 3D maps I showed you. We by now have retired our in-house protocol, because this modification of this commercial kit, of the ARIMA high C kit, gives us a much better, in general, better cis-trans ratios, and in general, higher percentage of artifact-free chimeric reads than our in-house protocol. And so that's, if you're interested in the transposase protocol, I mean, we can send it to you, and it's all documented in the genome medicine paper. At this point, I wouldn't recommend it as a first choice because it just has a poorer signal to noise in general, in part because of the lack of the biotinylation enrichment.
Excellent. Dan says, thank you very much.